My name is George Kraw. My wife Rafe and I have created this series in order to showcase people like Harry Noller and the wonderful, brilliant scientists at UC Santa Cruz to make more accessible to the public at large the important work that is being done there. Before I turn this over to the Chancellor for a few remarks, I'd like to recognize some of the representatives of public officials and public officials who are here tonight. I believe we have someone from Anna Eschew's office here. Yes. We also have we're supposed to have a representative from Ken Yeager's office. I don't know if they've made it here yet. And then we have a representative, I believe, from Representative Ro Khanna's office. And we have Assemblyman Ash Kawa here from the 27th District in the back. Maybe you could stand up for, appreciate you coming. So at this time, I'd like to turn it over to Chancellor Blumenthal for opening comments.
Thank you, George, and thank you all for being here. It's really a wonderful evening, and we are all honored to be able to have Harry Noller here, to hear about this, what all of this RNA stuff is all about, and what it means for our health, because I can tell you the news is good. There are exciting developments going on every front. You know, we're brought together tonight under the most delightful of circumstances because Harry won the 2017 Breakthrough Prize in the Life Sciences. I actually think it was the 2016 prize, but and that really is a big deal. Some call it the Nobel Prize of Silicon Valley. It was established, the prize was established in 2013 by a cohort of crusaders whose names are familiar to all of us. Names like Sergey Brin, Anne Wojcicki, Mark Zuckerberg and Priscilla Chan, and Yuri and Julia Milner. And they established the Breakthrough Prize to recognize great scientists, the people who are changing our fundamental understanding of the world. Each year they present an award for breakthrough knowledge in fundamental physics, life sciences, and mathematics. And candidates are nominated in a very open online process, but the recipients are all chosen by former laureates, by people who had already won the prize. And they emerge from their labs trading white coats for tuxedos and sequined gowns to walk the red carpet with stars like Morgan Freeman. It's an impressive gala. It's kind of the Academy Awards of scientific discovery. Which brings us to Harry. I want to tell you a little bit about the man. This brilliant scientist, this gifted researcher and generous mentor. But first I'll share an anecdote that to me speaks volumes. When I first contacted Harry to congratulate him on winning the Breakthrough Award, he responded in a very typical way for Harry by saying, well, I am happy for the campus. And I thought, gee, he was thinking about the campus, not about the honor that was bestowed upon him. And that's quintessential Harry. You know, we've all known brilliant people, often with egos to match, but that is not who Harry is. He is soft-spoken, focused, and he's deliberate in his thoughts and in his actions. And he's also multi-talented. Not everyone knows that. He is a superbly gifted jazz saxophonist. He's played with Chet Baker and the Randy Masters Group. And he's also a race car enthusiast. He owns a classic Maserati and a Ferrari. And I've heard rumors that at times he's been known to exceed the speed limit as well. Harry says his philosophy is work hard and play hard. And indeed, that's what he does. It's Harry's work that brings us together tonight. Harry's breakthroughs in molecular biology upended what we know about the mechanisms of cellular biology. Ribosomal DNA performs the key functions of translating DNA's genetic code and doing it and building the proteins of all living cells. Harry calls the ribosome the mothership of life, and with good reason. This work has profound implications for understanding both the origins of life and the mechanisms of disease, and that's our focus tonight. True to form, Harry wanted to share the spotlight tonight with his team. So we'll hear from Harry and from 3 members of the Center for Molecular Biology of RNA. Sorry. Big mistake. It's not the worst mistake I've ever made. The center is home to more than a dozen powerhouse researchers: Manny Ayres, Al Zahler, David Hausler, and rising star Angela Brooks. Until the Breakthrough Prize, I think it's fair to say that the RNA Center was one of the best kept secrets on the UC Santa Cruz campus, but no more. Their work ranks up there with our world-class astronomy program and our fundamental achievements in genomics, assembling the first full sequence of the human genome in the year 2000 and posting it online, thereby ensuring that humanity's blueprint would be accessible by everyone and accessible for free. Harry's work on RNA and the ribosome is among the signature contributions UC Santa Cruz has made to humanity. So it is with enormous pleasure that I will be able to introduce him. Let me say a few more things. As often the case with scientific breakthroughs, the more we discover, the more we realize that there's a lot more to be known. We see that in astronomy, and we see that in genomics, and we see that in RNA biology. And it's exciting because Harry's been responsible for so many of these breakthroughs. And he'll tell you that he's most excited about what he's working on today, even though he's already received these great awards and achievements. So the quest never stops. So tonight we're going to shine a light on Harry, a tremendous scientist. He's also a kind, generous leader who attracts incredible talent and builds amazing teams. And one last thought. Harry gives a lot of credit to UC Santa Cruz and the culture of openness and daring that led him to explore his own crazy ideas. He arrived on campus in 1968, just 3 years after we opened our doors as a campus. There were more cows on campus than there were buildings at the time. But what those early days meant for Harry was that there were no powerhouse scientists around who could tell him not to pursue his ideas, or who could dismiss his ideas or push back on his hunches. They didn't tell him he couldn't do what he wanted to do. Today we have plenty of powerhouse scientists on campus, but that spirit of openness is still alive and well today. And that's why great minds flourish at UC Santa Cruz. To me, Harry is a reminder of what's possible to be a towering figure in his field and to maintain one's curiosity around the world, about the world around them, to keep asking big questions and to keep pushing boundaries, to remain humble in the face of all that we do not yet know. So it is an honor to introduce Harry Noller.
Well, thank you, George, for that humbling introduction. And I would second the idea that Santa Cruz is a place for, as we see on the banners on campus now, for outside-the-box thinking. It was when I arrived, very early days with the cows that George mentioned, and the students had just moved out of the house trailers and into actual buildings, but it was an exciting time. It was sort of an adventure, And I felt that the spirit is still there. And I wanted to thank George Kraw, the other George of the evening, for his great support of the Kraw lectures. I think this is a wonderful opening for our UCSC community. So I'm going to talk about RNA and then about ribosomes, and then you'll hear from some members of the RNA Center about what they do. So I'll start with, I'm going to assume that no one knows anything about molecular biology or RNA at the beginning, which will make some of you happy, and there's a lot of expert scientists and molecular biologists in the audience, and I will just ask for their patience during that part. So what is RNA? Well, everybody knows about DNA. That's become a sort of common word in our everyday vocabulary. It's taken on many meanings. Talk about the DNA of the Golden State Warriors. But I should mention that DNA is considered the genetic material. It stores the genetic information in our chromosomes, and in fact, in all living things, on all living organisms. However, and this is a spoiler alert, in last Sunday's New York Times Sunday crossword, clue number 87 down is genetic material, and it's 3 letters. And the answer is RNA. So it's official. So besides carrying genetic information, RNA carries out many other functions in living cells, in all living cells. And we're learning more and more about what RNA does to this day. When I was a graduate student back in the very early days of molecular biology, they discovered messenger RNA, that, okay, you bring the information from DNA in the form of messenger RNA, and that gets translated into protein, as I'll explain. But since then, about every few years, a new whole class of RNA has been discovered, and that is still happening. It has not let up, and we'll get to that as well. And one of the classes of RNA is ribosomal RNA, and it's the main molecule of the ribosome. And it works with 2 other RNAs, the messenger RNA that I mentioned, and transfer RNA, to make proteins. So what is the ribosome? I call it the mothership of life because it's been with us since the beginning of life as we know it. It reads the genetic code. It synthesizes all proteins in all living things. It's the link between genotype, that is the genetic information, and phenotype, who we are. And finally, since it was there at the beginning, 3.5 billion years ago, roughly, are there clues in the ribosome to tell us where we came from, the origin of life? So why should we study ribosomes? Not only are they fundamental to life, making all proteins for all living cells, but bacterial ribosomes are also targets of many important antibiotics. And this has led to understanding the mechanisms of actions of antibiotics, and even creating new classes of antibiotics based on the ribosome. And they're believed to be a link to what we call the RNA world, which is where we believe we came from. So life is based on two molecular languages, the language of DNA and RNA. So the DNA language is G, A, C, and T. The RNA language is like a dialect or a closely related language, maybe like the difference between Spanish and Portuguese, say. So you have G, A, C, and U instead of G, A, C, and T. Let's see. Oh. The language of proteins is completely different, as you can see. You have, instead of these simple G, A, C, T, or these four-letter languages, you have a 20-letter language, ALIS, CIS, ASP, and so on. These are the amino acids. So G, A, C, and T are the nucleotides, and the protein letters are the amino acids. And so you have to go from the nucleic acid language to the protein language. And they are, as I say, as unrelated as English and Chinese. So ribosomes translate the RNA language into the protein language. So we have the genetic information stored in DNA, in our chromosomes, and gets translated into protein via RNA, so from genotype to phenotype. And this is what a DNA double helix looks like, and this is what typical proteins look like. And in between, you have RNA, which you can see has a double helical structure, or several kinds of double helical structures embedded in it that resemble the double helices of RNA. And so you go from here to here to here during translation. And the process of translation is heavily invested in RNA. So you have the messenger RNA, which is a transcript of the DNA that's exactly like the gene encoded in the DNA, except for changing T's to U's. And then you have two more molecules, the RNAs, the transfer RNA and ribosomal RNA, which play important roles. So the genetic code is the Rosetta Stone then between the very different languages of DNA or RNA and protein. And each amino acid is specified by 3 nucleotides or letters of RNA called a codon. And this is the genetic code, and it emerged in the early 1960s. There are 64 possible 3-letter words based on these 4 letters, and here they are. 3 of them, or 1 of them is a start codon, and 3 are stop codons, and the rest all code for different amino acids. So in the DNA, you have the coding for a gene as a series of 3-letter words, so TTC, TCA, CGT, and so on. And when they get transcribed into RNA, they become UUC, UCA, CGU, ACG, and so on. And then the ribosome then translates them into UUC becomes Phi, UCA becomes Cir, CGU becomes ARG, and so on. So the messenger RNA sitting in the ribosome looks like this. This is the actual structure of a messenger RNA sitting in the ribosome as we determined it in our lab at Santa Cruz. And so you have 3, this is 1 codon, this is another codon, this is another codon. This is the one that's currently being read. This is the one that was read before that, and this is the one that was read before that. So the messenger RNA is moving through the ribosome from right to left as we look at it there. This is the other RNA player, the transfer RNA, and it's sort of a bilingual molecule. At this end, it reads the messenger RNA codon. At this end, it brings in the amino acid. So it reads the message here, and the chemistry is done there. And it does the recognition using Watson-Crick base pairing, which is the same pairing that we see in DNA that is responsible for replication of DNA, that A always goes with T in DNA. In RNA, A always goes with U, and G always goes with C in both DNA and RNA. And this was discovered by Watson and Crick back in 1953 when they discovered the double helical structure of DNA. And in that, they proposed that A pairs with T and G pairs with C like this. And you can see that they are uniquely able to pair with each other by these dotted hydrogen bonds. And that's one of the key determinants of how life works. And in the ribosome, the tRNA does exactly this kind of base pairing between the transfer RNA and the messenger RNA. So here's a little short piece of messenger RNA. So here's one codon, here's another codon, and this one is being read, this yellow codon has 3 bases in it that are being read by 3 bases in the tRNA. So this is AU, AU, and GC in the, so the tRNA recognizes the codon by the same base pairing mechanism that Watson-Crick discovered in their structure of DNA. So ribosomes are very large. They're very complex in their 3-dimensional structure, and they're ancient. As I said before, they go back about 3.5 billion years. Their structural cores are virtually identical between all living things. So between our ribosomes, plant ribosomes, bacterial ribosomes, archaeal ribosomes, every living thing on our planet, if you go into the core, the functional core of the ribosome, you will not be able to tell whether you're in a bacterium or a human being. They contain, besides their RNA components, 50 different proteins, or in the case of our ribosomes, more than 70 different proteins, and very large, complex RNAs that make up 2/3 of their mass in bacteria, half of the mass in our ribosomes. An early experiment in our lab made us wonder whether the RNA was actually doing something. At the time I came to Santa Cruz, the common wisdom was that, well, the ribosome has 50 different proteins. Proteins are what do everything in life. They're the enzymes, the antibodies, make up our skin, our muscle, and so on, blood proteins. There were no ideas out there that anything but proteins carried out biological functions. But we did some experiments that seemed to rule out the idea that the proteins were important, and so we turned to the RNA. In this experiment that we published in 1972, myself and an undergraduate, bright, young, innocent undergraduate, Brad Chairs, Jonathan B stands for Bradford, and he went by Brad, and I published a paper saying that the ribosomal RNA was important for ribosome function. And, this This is Brad here with his sidekick, Jim Hogan, typical students of the time. So this led us into abandoning proteins, ribosomal proteins, as a pursuit, and we started functioning on the ribosomal RNA and thinking about the three-dimensional structure of the ribosome. And so where do you start? Well, the function of a biological molecule is determined by its three-dimensional structure, and we began by determining the primary structure of the 16S ribosomal RNA. That's one of the large RNAs of the ribosome. And with a brilliant postdoc from Germany, Jürgen Brosius, and two undergrads, Lindy Palmer and Poindexter Kennedy, we determined the first sequence of ribosomal RNA using actually DNA sequencing, and this was actually the longest DNA sequence ever sequenced at that time. So in this hippie commune at UCSC. And this is it. And do we have any sound there? No. Okay. Anyway, this was it. This is actually the sheet of paper that we first typed the whole sequence on from the different pieces back in 78. And so then the question was, how does this fold up into 3 dimensions? But that's the beginning. And because we knew that A's and U's base pair with each other and G's and C's base pair with each other, we had a hope of figuring out how this folded into 3 dimensions. But the first step was to figure out the base pairing. And this was in collaboration with Carl Woese at the University of Illinois. And he and I had a great collaboration going on for many years. And in the middle of that, Carl actually did his greatest work, the discovery of the third kingdom of life, the archaea, for which he received the Crawford Prize. But so this was the secondary structure of 16S ribosomal RNA, and each of these little sort of they look like maybe streets in a track development or something, is a double helix made out of RNA. And so the next problem is to figure out how these helices fold up in 3 dimensions. And so problem is that ribosomes are only a millionth of an inch in diameter, but at the same time, they were the largest asymmetric structure that anyone ever attempted to figure out the structure of. And so we decided to use X-ray crystallography, if we could, to see what that structure was. And so this is where it was when we started, and then electron microscopists began to see shapes, vague shapes, and then cryo-electron microscopy brought out more detail, and finally crystallography, we can now tell where every atom in the ribosome is. And this started with the arrival of 2 great Russian scientists, Marat Yusupov and his wife Gulnara Yusupova, who joined us in the late 90s to attempt to crystallize ribosomes. And we did obtain crystals, and these are what crystals of ribosomes look like. So one of the joys of crystallography is seeing these babies appear in the microscope. And then the idea now is, how do you get from a crystal to a 3-dimensional molecular structure? Well, in the crystal, the ribosomes are lined up in precise little rows in 3 dimensions. So in the X, Y, and Z dimensions, they're all sitting exactly in the same place. And so that means when you shine light of the correct wavelength on them, they will give diffraction patterns. And the correct wavelength of light turns out to be the very short wavelength of X-rays, which is has a wavelength of the order of the distance between atoms. And so we use an instrument called a synchrotron, which is about the size of a football field, to do this. And it creates the most intense X-rays that exist. This is the one at the Lawrence Berkeley National Laboratory where we took our first pictures. And so you put the crystal in this needle-sharp, intense beam of X-rays, and it shines on the crystal, producing a diffraction pattern with millions of spots, and they are captured on a detector, and they look like this. So this is one slice. You can imagine that the total data are actually a sphere of spots, and this is a slice through that sphere. So we take hundreds and hundreds of pictures like this, rotating the crystal by a fraction of a degree between snapshots, and then with the millions of spots, we measure the intensities of all the spots very carefully, and then using a mathematical procedure called a Fourier transform, we obtain the electron density in 3 dimensions, and that looks like this. So this is a close-up of part of the electron density map, and with that we're able to fit every atom in the ribosome where it belongs in the end. And so this then resulted in the 3-dimensional structure, and to show you how it happens, here's the secondary structure that we looked at with the different RNA helices in diagrammatic form, and here's the same thing in its actual 3-dimensional structure. So here, this yellow helix here is actually looks like this in real life. This red part here forms this head domain up here. The blue part and the magenta part form the body down here, and so on. That's the 16S RNA, and then we have the 23S RNA, and then of course 50 proteins besides that. And so that all makes up the mothership that we are now docking on. So this is a simplified view, and we'll look in a minute. It will morph to an all-atom view. This is now an all-atom view. So there are hundreds of thousands of atoms in the ribosome, and so it even overcooks the ability of the computer to display them. So this is a cross-section now. On the left is what we call the 30S subunit, on the right the 50S subunit. The messenger RNA is shown here in simplified form as this yellow tube, and it's being read by the tRNA here by base pairing, and then down here is where the chemistry happens that creates the protein, one amino acid at a time, and you see the nascent protein chain going through a tunnel in the 50S subunit, and it will eventually come out here and fold up into 3 dimensions and do whatever that protein is destined to do. So we'll look at an animation done by this brilliant animation artist in Australia, Drew Barry. It's not strictly accurate, but it will give you a picture of the process of protein synthesis. So the small subunit jumps on, and then the large subunit, and now oh, here we go. There's a great soundtrack to this. The these are the transfer RNAs coming in. This is the messenger RNA being read in groups of 3 nucleotides. One codon at a time. And now you see the tRNAs coming in, the nascent protein chain coming out. This is actually only a tenth as fast as the actual ribosome goes. These are different tRNAs with different amino acids attached. You'll see the protein chain starting to come out and then folding up into its 3-dimensional structure. Okay. So during this process, you can appreciate now that the ribosome is actually a molecular machine. So the tRNAs come in to the ribosome and read the message, which is the messenger RNAs wrapped around here. So they'll come in one at a time, reading the new codon, making a peptide bond, moving to the P site, moving to the E site, and then leaving. So they move from right to left, from A to P to E, and the message moves also, as shown in this diagram here. So how does this work? So it's a vastly complex molecular machine with moving parts. But by trapping the ribosome in intermediate states that we can crystallize, that states that will sit there long enough for crystals to form, which is from days to weeks, we can then solve the crystal structures of these trapped complexes, and then we can morph from one structure to the next to recreate the motion inside the ribosome. And this work was done by several talented scientists in our laboratory, J. Joe, Laura Lancaster, John Paul Donahue, and Sri Vidya Mohan. So first I'll just show you this motion as it happens on the 30S subunit, so just sort of half of the ribosome, but we can see everything here. These are 2 tRNAs, the A site and the P site tRNAs. So the object is to move this guy from here to here, and this guy from here to here, and then out. And so the ribosome does not simply say to this tRNA, good luck, I hope you make it to the P site. It's Mother Nature takes care of all her molecular children and says, you know, don't worry, I'm going to carry you from here to here, and then I'm going to take you to the next site, and then everything's going to be okay. Oops. So the first is the rotation of the body, and then a rotation of the head, and then a back rotation to reset. So the rotation of the entire 30S subunit, and now the head rotates, and now everything rotates back to reset, and the new tRNA comes in. We'll look from the top, and first you'll see it'll rotate this way, and now the head undergoes a very large-scale rotation, and then it moves back and resets. So these are very fundamental movements that go back to the origin of life. So this is some of the first motions that linked genotype with phenotype. And we'll see it in the context of the whole ribosome. First, the 30S subunit moves. In the background, you can see the 50S subunit, then the head rotation, reset, and now we'll view it from the top. You can see the 30S rock, and then the head rotation, along with movement of the tRNAs and the messenger RNA. We fade out the ribosome so you can see more clearly the tRNA is moving with the ribosome. And this is the messenger RNA here. You can see it move toward us now during this next rotational event. So we can look back now and see, we can see that everything is based on RNA in the ribosome, that it's three main functions: the selection of tRNA, the catalysis of formation of the bond between the amino acids, and this very complex movement are all properties of its RNA. So, as Francis Crick suggested in 1968, the same year I arrived at Santa Cruz, was the first ribosome made exclusively of RNA. So if we look at the basis of life, life as we know it is DNA makes RNA and RNA makes protein. But DNA most likely evolved from RNA as a more stable way to store genetic information. RNA is more fragile chemically, and DNA is pretty robust. So before the arrival of DNA, RNA would have stored the genetic information. And it still does today in fact, most viruses that cause human disease. So the common cold virus, poliovirus, rabies virus, HIV virus, are all RNA viruses. So these viruses store their genetic information as RNA. And so we believe what happened is it started with RNA because RNA can do everything that proteins can do, essentially, as well as store genetic information. And RNA was able to convert itself into DNA to store the genetic information. And this would have been an RNA protein world then, with RNA storing the genetic information. So in this RNA protein world, RNA makes RNA and RNA makes protein. But before that, we think there was an RNA world because RNA is able to carry out many of the biological functions previously thought to be exclusive to proteins, catalysis, and so on. So it's widely believed that life originated in an RNA world where RNA served the functions of both DNA and protein. So the RNA world would be simpler yet. RNA makes RNA makes RNA. So that takes us to the next order of business tonight, and that is the Center for Molecular Biology of RNA at UCSC. There are now 16 laboratories devoted to studying RNA biology. So we have the largest center for RNA research in the world, doing research ranging from RNA structure and function to RNA genomics, RNA medicine, and the origin of life. Department of Molecular, Cell, and Developmental Biology. We have 7 labs, and you'll hear from 3 of them. Our newest recruit, Joshua Aribere, Susan Carpenter, and Jeremy Sanford. In Department of Chemistry, we have 2 labs, And in the Department of Biomolecular Engineering, we have another 7, and one of whom, Daniel Kim, you'll hear from tonight as well. And we get to do it in this wonderful setting. And so this evening is sort of the launching of a new challenge, which is to understand all of RNA in our human cells. And we call it the dark matter of biology, and which our chancellor, being an astrophysicist, can relate to. So the human genome, which was assembled for the first time at UC Santa Cruz with the efforts of Jim Kent, David Hausler, and their colleagues, has 3 billion base pairs of DNA, but only 2% of that DNA codes for protein. More than 85% of it codes for RNA. We know the biological roles of only a tiny fraction of that 85%. But we already know that it includes RNAs that are crucial for regulation of gene expression, some defects in which are responsible for human disease, including cancer. So an enormous challenge for the future is to identify the RNAs that make up our dark matter and to understand their biology down to the atomic level. So we have an incredible group of scientists who are uniquely capable of attacking this whole problem. So we're now going to hear 3 very exciting stories from RNA Center faculty, Jeremy Sanford, Susan Carpenter, and Daniel Kim, of how discoveries from basic research into the biology of RNA are already opening the door to new approaches to diagnosis and cures for human disease, including cancer. You'll hear about important classes of RNA that were completely unknown only a few years ago. This will be followed by a roundtable discussion in which we'll be joined by Joshua Araberi, who has just joined us from Sanford University, as our newest RNA Center faculty. And we'll invite you to join us in the open question and answer discussion at the end. So I'll now turn it over to Jeremy Sanford.
Okay. Awesome. Can I borrow your pointer? Thanks. All right. Okay. Let me remember what I got to do. Okay. I'm going the wrong way. All right. Okay. So it's a real pleasure to be here with you all tonight, and it's fantastic to be able to just spend a little bit of time to tell you about the work that's going on in our laboratory. And actually, when I got this opportunity, I thought, I'm going to tell everyone all about everything that we're doing, and I tried, and it was a miserable failure. So instead, I thought I'd just tell you about one of the projects that we're really excited about, because we think it illuminates some new roles for RNA in cancer. But before, I think I'm going to start off with a somewhat heretical statement. And so, Harry, I hope you don't take my card, okay? But, there are some proteins that are interesting, right? and I'm going to preface that by saying they happen to be the proteins that bind to RNA, okay? And one of the things that we're really interested in our lab, as Harry described these 2 molecules made up of 2 chemically distinct languages. And in our laboratory, we're really interested in this intimate dialogue that occurs between proteins and RNA. And one of the things that Harry mentioned in the central dogma, that flow of genetic information from DNA to RNA to protein, he described that translation process as being heavily invested in RNA, right? And And that's a really important and profound statement. But the human genome is heavily invested in proteins that bind to RNA. So out of those 20,000 genes, there's something like 1,500 that are devoted towards managing RNA, right? About 170 are related in some way or another to translation, to the ribosome, to prepping tRNAs for their job. And another 800 or so are involved in binding to the messenger RNA, and that's what my lab studies. So what I'm showing you here is just an example of the type of dialogue that I'm thinking about when I say protein-RNA interactions. And so what you've got here is this beautiful sort of banana slug-like piece of RNA, and it's being sort of warmly embraced by a protein. And you can see just how many contacts there are between the RNA and the protein. And you can imagine that if something goes wrong with this interaction, it could undermine functions in the cell. And this is really critical in the context of the flow of genetic information, where these RNA binding proteins play massive roles in managing the messenger RNA. So what I'm going to tell you about today is an RNA binding protein that we got really excited about because we think it has an important function in cancer. And so this RNA binding protein is called IMP3, IMP3. And I think if you could remember just one thing about the talk, it would be that this protein is a really devious protein, right? If it gets turned on in the wrong cell at the wrong time, it can massively increase the malignancy of cancer, so it can be much more lethal. So it really is kind of a devilish protein. But it turns out that it's interesting for many other reasons too. So the first thing that I want to tell you about IMP3 is that normally it does its job during early embryonic development. And so what I'm showing you here is a mouse embryo that's been stained in order to reveal the active IMP3 gene. And so all this dark matter here, this dark signal, turns out to be places where the IMP3 protein is made. And you can see they're in important regions like the brain and the eye, developing limbs. These are tissues full of cells that are proliferating rapidly, and they're undergoing morphogenesis. What's really fascinating about this gene is that as soon as it's switched on, just a few days later, it's switched back off again. And what you can see in this incredibly boring slide is that if you were to survey 50 different tissues from healthy adults, and so this experiment encompasses more than 70 samples from of these different tissues, so 70 different people contributed these tissues. There's absolutely no expression, no evidence for the activation of this gene at all, right? So it's pretty much, you've got a gene that's on in a very narrow developmental window, and then it's switched off all the way through adulthood. But the story gets a little bit more interesting when you look in cancerous tissue. And so what I'm showing you here are biopsies from either a healthy individual's pancreas or patients that have either low-grade or high-grade pancreatic cancer. And what you can see is that in comparison to this nice clear field of pancreatic cells, as you go from low-grade to high-grade, you can see there's much more of this brown staining pattern. And that brown pattern corresponds to signal from staining these cells with antibodies that recognize the IMP3 protein. So you can see that in high-grade cancers, there's more IMP3 protein, and that's fairly interesting. And so it turns out that this isn't just restricted to pancreatic cancer. Numerous people have reported that IMP3 is highly upregulated in advanced tumors of a wide array of types: breast cancer, colon cancer, brain cancer, you name it. And so it was pretty exciting to me when a few years ago, a really great friend of mine named Dinesh Rao, who's a pathologist at UCLA, I've known him for about 25 years. We were undergraduates together. He was in my wedding. He's a fantastic guy. And what's interesting about him is he's an MD PhD, and he studies pediatric leukemias. And he is also interested in RNA-based mechanisms of disease. And so he called me up one day to tell me that, hey, he was looking at patients who came into his clinic with 3 different types of leukemias. And he clusters them in this picture. So each row is a different patient. Each column is a different RNA binding protein gene. And what he told me, because he knew I was working on IMP3, or at least thinking about working on IMP3, and he said, the patients that have the most severe form of leukemia, this MLL-driven leukemia, have the greatest abundance of the IMP3 gene. It's massively induced in these patients, and it's more or less missing in the lesser forms. And so he did a little bit more homework, and we made these plots here which compare patient survival in both leukemia and pancreatic cancer. And what we did was divide these patients up into two different cohorts. Those that have very low or no expression of IMP3, shown in green or blue, and those that have really high expression of IMP3, shown in red or orange. And this is expression in their tumors. And what I think almost anyone can see is that there's a big difference in the shape of these curves. And so what you're looking at is the fraction of patients that survive after a certain amount of time. And you can clearly see that if you have elevated IMP3 levels, there's this clear correlation with patient survival. And so all these observations served sort of the premise for the experiments that I'm going to tell you about. And this is the real question that Dinesh and I wanted to go after. So is IMP3 a cancer driver? It was clear from the literature and from our other studies that many people thought IMP3 was a biomarker, simply something that's correlated with the progression of the disease. And why is that? Because IMP3 really has only a couple of telltale features. All of those features are related to RNA. And the people who study cancer, they don't think about RNA. So we got excited about this. And so we did a number of experiments to distinguish between these two possibilities. It's either a driver or it's a passenger, right? And I'm going to tell you about just 3 of them. And this is the most important experiment or a result that we got. And the idea was that we wanted to know if we turned the IMP3 gene on in a mouse in the wrong cell type at the wrong time, would that be sufficient to cause cancer? And so we created a system where we could turn IMP3 on in the hematopoietic system. So these are the bone marrow stem cells that give rise to all of your blood. And this is the system that normally fails in leukemia. And what we discovered is that when we turned IMP3 on in these cells, that we were able to induce essentially leukemia in these mice. We could see all of the telltale signs of leukemia. And that was really exciting because that was the first time that anyone had demonstrated that this was really a pathological player in disease in an animal model. So that was cool. We were pretty excited. And then we did 2 more experiments, and these were a little bit less sophisticated. The idea was that we took pancreatic cancer cells or human leukemia cells, grew them in the lab, and we took away IMP3 from those cells. And what we saw was that the pancreatic cancer cells were far less invasive, they were much less aggressive, and the leukemia cells were much less proliferative. So we basically shut down the growth of those cells. And so that really told us together that we think IMP3 probably is a driver, it probably is a key player in the development or the progression of cancer. And so it also suggested to us right away that because this thing interacts with RNA, that maybe there's a novel strategy that we could take to treat cancer, which is to target the RNA. So maybe we could develop some new drugs to really interfere with that dialogue between IMP3 and its RNA targets. All right, so, but it's a big challenge, right? And one of the challenges are that we don't really know what IMP3 is doing in the cell. I can't show you a beautiful movie of IMP3 grabbing onto things and interacting with other things. We just don't know what it does, okay? And we have some hunches based on some stuff that I'll show you in a second. And then the other really important part of this is we don't know what RNA molecules, or we didn't until recently, that this IMP3 protein is interacting with. What's it talking to? And just to give you a scope of that last problem, I'm going to reiterate the idea that Harry said, the human genome has 3 billion bases. And I rounded up because the math is easier for me. But I'm going to assume that about 90% of this is transcribed into RNA. And that means we've got about 2.7 billion bits of information that correspond to positions in RNA where IMP3 could be interacting and doing a job that's important for cancer. And so the question is, how do we find those key places? That's like a giant RNA haystack, and we have to go find these little RNA needles. So fortunately, my lab has really tremendous expertise at finding these RNA needles in these giant haystacks of nucleic acids. And to do that, we have an approach where we can really just reach into the cancer cell with an antibody. We can use the antibody to grab that protein, in this case IMP3, pull it out, and that brings with it RNAs that it's directly associating with, okay? And then that leaves behind all of the rest of the RNA that's in that haystack. And from there, we can sequence this information, compare it to the human genome, and we generate these things that we call RNA maps. And so the idea is that this map is going to point us to the positions where we think IMP3 is doing a job, right? And that allows us to really answer, I think in some ways, begin to answer both of these questions. So I'm going to show you two of the maps that we've made in leukemia cells and pancreatic cancers. Again, not as exciting as the ribosome moving, but what I want you to take note of is just that these maps, which show where the protein likes to interact on RNA, this region is the region where the ribosome does its job, this region is the regulatory region. We can see that IMP3 seems to be in this region of the message, and both of these maps in leukemia and pancreatic cancer cells look very similar. And if you zoom in on a particular region, this is what we got excited about. We found a remarkable overlap between where IMP3, this little volcano type thing here, where IMP3 likes to bind, so it sticks to mRNA right near this 0 position. And this 0 position turns out to be places where an important class of RNA regulators interact with messages. And these are called microRNAs. Now, microRNAs are really fascinating because they're tiny relative to a message, only 22 nucleotides long. But yet they play huge roles in regulating the way our genes are turned on and turned off. And so this suggested to us right away, maybe IMP3 is collaborating with microRNAs to do its job, right? And so, we've definitely done some experiments to test that hypothesis, and it's still ongoing, but we think that's probably the case. So the picture that I want to leave you with is kind of our model. We imagine that when IMP3 is turned on in a cancer cell, when it goes rogue, right? It's going to be able to find RNA molecules, messages that that are made up out of this big, are composed this big RNA haystack, and these messages are going to play key roles in regulating the proliferation of the cell, so cell growth, the migration of the motility, and the cell adhesive properties of the cell. These are attributes that are really similar to what IMP3 might do during development when all those changes in the embryo are taking place. At the molecular level, we imagine, and we have evidence, that IMP3, through its interaction with the message, right, may work together with these microRNAs. And so what we're really excited about is the idea that if we could sort of interrupt this dialogue between IMP3 and its RNA targets, or the interactions between IMP3 and the microRNA machinery, that we could really create new treatments for cancer by interfering with protein-RNA interactions. And this would be a massive departure for the rest of the field. If we were to develop RNA-based drugs, this is something the rest of the cancer field in general is really focused on targeting proteins that we know a lot about, enzymes that have active sites, we have the three-dimensional shape, we can find a wrench to stick in there. And the problem is that proteins often find ways around that, and so you get remission. We imagine if we interfere with IMP3, we can affect the expression of hundreds of genes simultaneously. So I'll just leave you with that, and I guess when I think about this project, the last thing I guess I'll say is that we started this really just 3 or 4 years ago, and we've come a really long way. And why did we get here so fast? We've identified key targets, we have a possible mechanism in mind, we've got animal models that we're working on. And I think part of it is that I'm part of this fantastic group of researchers. And did you guys just cut me off? No. It's a fantastic group of researchers. And there's this amazing tradition of tackling the hardest problems in RNA biology at UC Santa Cruz. And I think we're going to keep doing that. So with that, I'm going to invite my colleague, Susan Carpenter, to the stage. And she's really one of the newest members of the RNA Center. She's been here for about 2 years. And she's working on one of these really awesome, mysterious classes of RNAs.
Thanks, Jeremy. So I'm an immunologist, and one of my main motivations to move to UCSC was to become part of the RNA Center, and not just because it's a place that embraces crazy ideas, but also because it's home to some of the world's best RNA biologists that I really want to work alongside. Because my lab is very interested in understanding how RNA can work to control our immune system. And so tonight I'm going to tell you about our work on harnessing these genes that we call long non-coding RNAs, or link RNAs for short, to diagnose and treat inflammatory disorders. So my lab is interested in the process of inflammation, something that I'm sure everyone here is familiar with. The process involves redness, heat, and swelling. And this is an essential process in your body to keep you healthy. So you need inflammation to occur to protect you from infection, from bacteria and viruses. But we're very interested in what happens if inflammation doesn't get turned off properly. Because if inflammation is not controlled properly, it can lead to chronic inflammation. And chronic inflammation, as we know, really lies at the heart of nearly every disorder you can think of, including metabolic disorders such as diabetes, heart disease, neurological disorders including Alzheimer's, autoimmunity, cancer, and my lab is particularly interested in chronic inflammatory disorders that includes rheumatoid arthritis and lupus. And what we're interested in knowing is how RNA is involved in controlling chronic inflammation. Because our idea is if we can get a better handle on how chronic inflammation occurs, perhaps we can develop better therapeutics to target chronic inflammation. And in doing so, we could develop drugs that could impact a huge number of different diseases. So the mission of my lab is to identify new genes that control inflammation. And we're very interested in developing biomarkers and new therapeutic targets for inflammatory diseases such as rheumatoid arthritis. Because these disorders are extremely difficult to diagnose at the moment. It can take up to a year to be diagnosed with arthritis, and you can imagine in that time a lot of damage has occurred. So we're very eager to develop much more rapid ways of diagnosing these diseases. So how do we go about doing this? Well, Harry introduced you to the central dogma of how inflammation flows through the cell, such that DNA makes RNA to make protein. But as he mentioned, there's a lot of dark matter in the genome. And we're very interested in understanding this dark matter because up to now, we have really all been protein-centric biologists. We have all studied proteins, and yet we haven't cured diseases such as arthritis. So we're interested in understanding how RNA is involved in this. And what Harry introduced you to is the fact that less than 3% of our genome actually makes proteins, while the majority of the genome gets transcribed into RNA. So we're interested in knowing what is all this RNA doing, and is it all biologically relevant? And how I like to think about this is, so far to date, we've all been studying proteins, and really they're just the tip of the iceberg. And we're really interested in understanding what's going on below here in the genes that are not making protein, but are in fact functioning as RNAs. And we're interested in studying the largest group of RNA that gets made in your genome, and they are called long non-coding RNAs, lincRNAs. So what are lincRNAs? They are genes that function as RNA, they do not make protein. They're defined as any transcript that's greater than 200 base pairs long. Imagine in our human genome, we have now identified 16,000 lincRNAs, and yet we know the function for less than 1% of these genes. And they're a particularly fascinating group of RNA for a number of reasons. And one of them that I think is particularly striking is the fact that lincRNA number increases with evolutionary time. So humans possess the most lincRNAs out of all species. Whereas if we think of on the level of proteins, we as humans do not differ in any way to a worm or a fly or a mouse in terms of our protein repertoire, yet we differ wildly when it comes to the lincRNA numbers that are expressed. So we like to hypothesize that perhaps lincRNAs are what make us human. And so my lab is particularly interested in understanding how these lincRNA genes are involved in controlling inflammation. And how we do this is that we can take blood from healthy donors. We isolate our immune cells, such as macrophages, that are the critical cells to help protect you against infection. And we can turn on inflammation in these cells and then ask what lincRNAs are getting turned on and which get turned off through sequencing. And this is just an example of that data as a volcano plot. And each of these little dots represents a different lincRNA. And what I'm showing you is that a large number of lincRNAs get turned on following inflammation, and a large number of them get turned off. But how do we figure out which of these dots are most critical for controlling inflammation? And to do this, we have developed a high-throughput system using CRISPR technology. So CRISPR really has revolutionized genome editing. As I'm sure you've heard, it's been pretty popular in the press for a number of reasons. But we're particularly excited to use this technology, and this flow-through of a workflow was developed while I was at UCSF as a postdoctoral trainee. And we are using this high-throughput approach to ask what lincRNAs can control inflammation. And how we can do this is that I have developed these reporter cells over here that we can turn on inflammation, and by turning on inflammation, you turn the cells green. And this is an example of what they actually look like under the scope. And then we can simply remove lincRNAs and ask which of them can turn off inflammation by turning off the level of green in the cells. And the power of this technology is that we can do this and look at all 16,000 lincRNAs at the exact same time. And so using this technology, we'll be able to study all lincRNAs in parallel and ask which of these are critical to control inflammation. And so really what we have using this technology, we can rapidly speed up how we discover which genes are important. Up until now, we've only been able to study these one at a time. So you can imagine for 16,000 genes, that would take decades and hundreds of labs. Whereas using this approach, we can identify the critical genes in a matter of months. And then by identifying these genes that control inflammation, we can then use these as potential targets for therapies. Another feature that my lab is working on in terms of lincRNAs are developing biomarkers to diagnose rheumatoid arthritis. And why we're interested in this is that lincRNAs have a very interesting feature in that their expression patterns are highly specific. They're even more specific than proteins. And what this means is that you have a signature of lincRNAs that are specifically expressed in every organ of your body and every cell type. And we also hypothesize that this is the case in terms of disease. And so what we are now doing is we can take blood from healthy donors and patients with arthritis, simply take the blood and sequence them to identify the genes that are specific only to arthritis patients. And in doing so, this will enable us to develop a very simple PCR-based diagnostic test for arthritis, and allowing for a much more rapid diagnosis. So in conclusion, what I've shown you tonight is the mission of my lab is to identify new genes, lincRNAs that can control inflammation, and we then want to use these as new therapeutic targets for arthritis. And finally, we are developing biomarker assays to allow for rapid diagnosis of rheumatoid arthritis using a very simple blood-based assay, so that you can go from what typically has taken over a year to diagnose, we can do it in a matter of hours, is the hope. And with that, I will introduce Daniel Kim, who is going to continue the theme of lincRNAs, at this time in cancer. Thank you.
Okay, thank you, Susan, and thank you to everyone for coming. It's really a pleasure to be here this evening with you and to share about some of the work that our lab is doing to help illuminate cancer through studying this RNA dark matter. And so, you've heard this term dark matter, and what is dark matter? I was actually Googling dark matter the other day, and I came across one of our chancellor's papers from 1984, and he describes very elegantly, real dark matter in outer space. That basically, if I got the gist of it, it's just matter that's really, really dark. Like you can't see it, so it's just invisible. But the good thing about RNA dark matter is that it's not invisible. We can actually see this dark matter. And so imagine if you took the Hubble telescope and you could shrink it down to the size of a cell, and you could peer inside of a cell. This is actually what you would see. This is not the night sky. These aren't stars in the night sky. These are actually RNA molecules, individual RNAs within a single cell. So you can actually see the border of the cell, and you can see these individual RNAs that I've lit up with this fluorescent tag. And so we have these amazing technologies now where we could see RNA actually in a cell. And we could do this, for example, for one gene. And so for this cell here, I've only labeled all the RNAs that are coming from one single gene. And already you could see dozens of these RNA molecules. But we can also do this using a really super cool technique called using different colors to look at multiple genes. And so here I've labeled 4 different genes with different colors. And so you can see, for example, this blue RNA, you can see a yellow RNA, you can see a red RNA. And so you can begin to see the complexity, even just from 4 genes, the RNA that's that's produced from 4 genes, you can begin to appreciate within a single cell how complex this RNA world is. And so, as we've heard very elegantly from Susan and from Jeremy and from Harry, there's a lot of complexity in this RNA world. And so, we know that there are 20,000 protein coding genes that are all making RNAs. We know that there are 16,000 of these link RNA genes that are all making RNAs. And so really what we need is a way to look at all of these RNAs all at once to really figure out, what are they doing together in the cell. And so the good news is we can actually do this. We have these amazing genomic technologies that have been developed in recent years to look at all the RNAs that are found within a single cell. And we're really fortunate to have some of the world's leading genomics researchers at UC Santa Cruz, such as David Hausler and others, who have really pioneered these technologies and these approaches to analyze this genomic data. And so what I'm showing you here is just, again, the landscape of all the RNAs that are expressed within this single cell, red indicating high levels of RNA and blue indicating low levels. And so, as Susan described, these link RNAs are really this mysterious new class of RNA dark matter that our lab is really interested in studying. And so perhaps the most salient feature of these link RNAs is that they have this exquisite specificity in terms of where they're expressed in the body. So, for example, if I just gave you a chunk of DNA from my body, you wouldn't know where in the body this DNA came from, since all the cells in our body essentially have the same DNA. But if I gave you, for example, a link RNA, you'd be able to say, oh, this RNA is found normally in the pancreas. And so, in a way, it's an RNA-based IP address, if you will, where you could see, where is this RNA coming from, and you can really leverage that information to help diagnose and treat diseases, which I'll tell you about in just a minute. And so, when I began my lab at UC Santa Cruz about a little less than a year ago, as Susan mentioned, I was really excited about all of the wonderful RNA scientists and researchers that were at UC Santa Cruz. And I really wanted to kind of focus the lab on one particular disease, cancer. And in particular, I wanted to focus on one gene that's really important in cancer. And so these RAS genes, which you may have heard of, are probably the most significantly mutated genes in all of human cancers. And so more than 30% of human cancers have mutations in these RAS genes. And so I'm sure everyone in this room has been touched by cancer in some way. And we're really passionate about trying to find ways to help to treat and cure these diseases, such as pancreatic cancer, which has an abysmal 5-year survival rate. Only 3 out of 100 people will survive this diagnosis of pancreatic cancer. But diseases such as pancreatic cancer, colorectal cancer, and lung cancer are driven by this mutation in RAS. And so, you've heard from the talks this evening that we have these 3 billion letters in our genome. And so what happens is there's a single mutation in one letter in one gene that can initiate this process of cancer formation. And so these are called oncogenes, these genes that drive the formation of cancer. And so our lab is really interested in studying how this simple mutation that initiates cancer in our bodies is affecting all of the RNAs within our cells. And so we wanted to just ask this simple question: how do RNAs respond to this RAS mutation in cancer? And more specifically, how do all of the RNAs respond, including the RNA dark matter? So if you have some landscape of RNAs that are expressed within, for example, a lung cell, and if you introduce just one change in the DNA, one mutation in one letter of one gene, how does that affect the landscape of RNAs that are expressed in this cell that is becoming cancerous? And so what we did, I'll just show you one quick experiment, is we basically took normal lung cells and we introduced this one single mutation in RAS, and we used these genomic technologies to sequence all of the RNAs that were being turned on or turned off by this simple mutation. And so what you see in red is all these dots indicating RNAs that are turned on during cancer, and blue indicates RNAs that are being turned off. And so what we're trying to do is we're trying to kind of cut through the noise and kind of look at these highly specific link RNAs. And so as you can see, instead of those hundreds of genes, now we have this more defined signature, this highly specific link RNA signature that represents this mutation in RAS. And so these link RNAs, as we've been kind of saying over and over, is they're really important because they have this exquisite specificity of expression. And so if we took any of these link RNAs and we found it somewhere in the body, we could confidently say these RNAs have been turned on by this specific mutation in RAS, and that they're found in lung cells only. They're not found in the pancreas, they're not found in the liver, they're not found in the kidney. And so this highly specific RNA signature then allows us to think about ways that we could diagnose and potentially cure cancers. And so what my lab is really interested in doing right now is creating this lincRNA atlas, looking at all the different RNAs that are turning on as cancer forms within the body. And so we're really interested in these early RNAs that are turning on, these early RNA signatures that could indicate that there's a mutation, for example, in the lung or in the pancreas, in this RAS gene that's driving the formation of cancer. And so really, I think the hope and the vision for our lab is really to develop this precision cancer detection method through basically a simple blood test. And so I'm sure many of you have heard of this term liquid biopsy. It's really all the rage in the cancer field right now, where people are just simply taking a little bit of blood from a patient and they're able to detect mutations in the DNA. And so when you sequence the DNA that's kind of floating around in these liquid biopsies that are circulating throughout your bodies that have been emitted by these cancer cells, you can actually see that there is, for example, a mutation, for example, in the RAS gene. But while the whole world is kind of focused on the DNA, again, at UC Santa Cruz, we're really embracing this non-traditional form of thinking and really embracing this RNA world. And so what I hope I've convinced you of is that the RNA contains more information, going back to this idea of an RNA-based IP address, where you can not only know that there is a mutation within, for example, the RAS gene, but you can also know where in the body this mutation came from, for example, in the pancreas or in the lung. And so what we're really trying to do is we're trying to make these fundamental discoveries looking at lincRNAs that are responding to, for example, RAS mutation, and then using that information to help diagnose disease. And so what I hope I've convinced you of today is that really through these fundamental basic science discoveries, where we're trying to study how, for example, a simple mutation affects all the RNAs within our cells, we can begin to leverage this information to really begin to try to cure diseases. And so really by charting this dark matter, this RNA dark matter, we really hope to illuminate diseases such as cancer, really leveraging the unique properties of RNA, and also through this collaborative effort of these 16 amazing researchers, these RNA researchers at UC Santa Cruz. And really, it's all because of the vision of our fearless leader, Harry Noller, who has really made this possible, bringing together these wonderful RNA labs to really help cure diseases such as cancer. And so thank you to Harry, and thank you everyone for coming tonight. Thank you.
All right, so I guess we're gonna spend the rest of the time sort of just having a discussion about RNA and genes and maybe even a little bit of health. And one of the people that we'd like to introduce is really this year's top draft pick, Joshua Arabari, coming from Sanford University, 5'10". But he's I'm going to keep going with this. He's a 5'2" player, really a prolific scientist, and we're thrilled that he's joined us. And so, Josh, why don't you come up and join us? And we've got Susan and Harry and Daniel coming up. All right, so I think I'm gonna start just by asking Josh. So Josh, what were you? You must be excited to be joining UCSC. What are you planning on doing? When you arrive.
Hiring someone first.
Yeah.
If anyone wants a job here. The focus of my lab is around quality control. You saw a lot of the ribosome acting well in translation. Things for the most part do go pretty well, but some fraction of the time they don't. And it turns out, in addition to the tRNAs flying around, there's also machinery very tightly watching that to make sure that when a mistake occurs, the cell can deal with that properly. And so I'm looking at how those quality control processes work, and sometimes this how the cell might turn on itself and degrade and get rid of something inappropriately.
Fantastic. Shoot. We have a question over here. Oh, that's okay. We got it.
So this is a question for the last speaker. You're looking at lincRNA in liquid biopsy type applications. No one has mentioned exosomes, so I'm wondering what's going on in the RNA group around exosomes.
Yeah, that's, am I still on? Yeah, can you guys hear me? Yeah, so that's an excellent question, thank you. And so if you actually look to see the the coverage of the RNA, the link RNA molecule. You can actually sequence little fragments of the RNA. And if you go and look at where the sequencing reads lie, you actually see full coverage of the RNA. So there's actually intact RNA that's circulating through the blood. And so most likely it is protected from degradation in the blood by some sort of microvesicle such as an exosome. So there's actually a lot of research that's really focusing on exosomes and how they might be involved in cancer and how they could kind of remodel the tumor microenvironment. And so one of the possibilities is that RNA is being delivered to these other cells around the tumor and kind of causing this reprogramming of the microenvironment.
I might actually add to that as well. Because in terms of our interest in making a diagnostic, we right now don't really mind or care where the RNA is as long as we can identify it. So being able to sequence this total RNA, we don't mind if it's in a microvesicle or it's being protected by a protein floating around the body. The idea is to make the simplest tests that will allow you to identify the RNA.
Thank you for great talks. I have a question for Professor Noller. Why is the magic number for codons 3?
That's a great question, and that's something that the founders of molecular biology discussed at length back in the day. And the first thing they noticed was there were a lot more amino acids than nucleotides. There are 4 nucleotides but 20 amino acids. So
Sure.
they said, well, if you had 2, that would give you, that is taking 4 nucleotides 2 ways, that gives you 16 possible codons. But from what the protein chemists had learned up to that point, there were actually 20 amino acids and possibly more. So that they decided that 3 was probably the minimum. But then that was unsatisfying. These guys were physicists, right? And so they didn't like the messiness of biology. And it, so it turns out, you need some amino acids more than others. There are a lot of alanines, but not many methionines and tryptophanes. So you have 1 codon for methionine, 1 codon for tryptophan, but 4 for alanine, 6 for leucine, and so on. The more common amino acids, you have multiple codons. So it's degenerate, as mathematicians would say, when, the square root of 4 is 2, or it can be minus 2. So, degenerate is, not a kind word, but that was the physicists dealing with biology. So 3, is the minimum number where you can specify at least 20 amino acids. It's also, there's a physical reason for that, and that is that the stability of reading 3 nucleotides is close to stable at biological temperatures. 2 would not be stable enough, so that's probably an important reason.
Harry, you remember when I was in your lab, we talked about the applications of computer to biology, and this was in 1970 when that was kind of a crazy idea. I know now it's very much
You were,
computerized.
as I remember, using the IBM 36040 in the basement of communications. We had all the power of my Seiko wristwatch.
And what we were interested in was discovering the tertiary structure. In that time, I was as indoctrinated as everybody else of proteins. It seems to me, first of all, that to understand these RNAs, we're going to have to understand their tertiary structure, so we'll know what the heck we're doing. I'm wondering, first of all, has there been progress made in being able to solve that purely by computer simulation? And then, for extra credit, is the advent of potential quantum computing anything that might be applicable to this?
Well, why don't you come back to the lab, Dave? We'll work this out. Those are great questions. So the folding of RNA has had a rocky history, but I think it's finally at the point where you can accurately predict the secondary structure, but we're still stuck at the three-dimensional, the tertiary folding in most cases. But particularly if you have something like ribosomal RNA where you have organisms with a lot of mutations with respect to each other, you can now find compensating mutations. So when you see a GC pair change to an AU pair, that gives you evidence that that's a real base pair. So that was the approach that we took with Carl Woese. But I think there's ways of getting at the three-dimensional folding by using semi-empirical methods based on the huge wealth of information on 3D RNA structure that we have just in the ribosome alone. So whenever you're ready.
So Harry, a few months ago you gave a talk at Santa Cruz, and you mentioned that in the ribosome, one or the other base will oftentimes, or not oftentimes, but vary throughout of all the biota. And there were, however, 15 in a row that, as if I understand it, I may have hallucinated when you're talking, but that 15 in a row have never changed in all time. And I thought at that point, I'll bet he's thought about what that means or what those 15 bases are about. Did I get that right?
Yeah, well, it's even, Dia Mohan, who's here, and I have been discussing this a lot in the last months. There are a couple of hundred of invariant nucleotides, or more than that, probably in the ribosome. That is, no organism has ever changed those in 3.5 billion years. So there's a death penalty, obviously, for doing that. And so I think what I would say is it's a measure of how well we understand the ribosome is how many of those can we explain. And I think at this point we can maybe account for 20 of them. So we're maybe 10% of the way of understanding the ribosome.
Hi. I'd like to thank everyone for the wonderful presentations. It was very informative. But I do have a question. I'm interested in the crossover between academia and research and the real world, I guess, the people that are trying to make the drugs to solve some of the problems you're talking about. How is the information passed? Are you in the process of trying to find these drugs yourselves, or is that something that's done outside of academia?
That's a great question. So typically, research labs do a lot of the grunt work. So we do a lot of the basic research, which we can then develop into patents that can be licensed to industry. But also one of the great things about being in Santa Cruz is that we do have some industry there and we can work very closely together. So my lab, for example, works with Somagenics, which is just down the road from us in Santa Cruz, and we're working together on these idea of trying to look at what genes are floating around in patient samples. And so typically we can either partner with companies that are interested in developing this technology, and for a lot of companies they've actually shut down their R&D, so they're not doing the necessary basic research to actually develop new drugs. And so that's where the academic labs take up that slack and actually, do the hard work. And so, we're very active with working both ways, both focusing in our own labs, but also doing outreach to try and move these things along more quickly.
Actually, that was a great question. I have a question that would dovetail on that. What are your challenges with funding, especially federal funding?
Yes.
Anyone can answer that question.
Federal funding is always a challenge. And it's been a challenge for years. So there really has been no increase in funding for research over the last 15 years. So this is not a new problem. And for a lot of what we're really excited about doing, which is, you know, as we said, the unconventional thinking, really isn't rewarded by funding through government agencies. So unfortunately, government funding really promotes safe research. They want to know that there's going to be a result nearly before, you've already done half the work before you get the money. And so we really do rely on, more philanthropy to allow us to do this much more high risk, high reward type research. And so, we work on both ways. We get our government funding to do our more developed projects, but we do require a lot of assistance from people who are willing to take a risk on something new to get this type of research done.
So what level of support do you need to get from where you are now to where you want to be? That's the bottom line question.
Well, if you...
We the entire panel.
I mean, what we're facing here is to answer this question, the dark matter challenge, with these 16 labs and probably more who will join us in the coming years, is, say, a 10-year horizon of attacking this huge global problem. What is all of the RNA in a human doing? And what are its implications for health and disease and evolution and everything else? Something like, to put a large round number out, would be something like $50 million over 10 years, which, going from 16 to 20 labs, something like that.
I mean, is this on? Yeah. So a big part of that, I think, is trying to support a lot of the facilities that we need to develop at UC Santa Cruz to really tackle this problem as well. We obviously have expertise in genomics, and we have experts like Susan who are applying CRISPR to lots of different problems. But there's a number of new skills that we want to bring into the university to solve these problems, and I think that's going to take considerable support to develop. But once we have that, we've talked about cryo-electron microscopy as being one of the key drivers of understanding the shape of molecules. It's almost outpacing crystallography as the preferred method for elucidating the three-dimensional shapes of molecules. And that's instrumentation that comes with a really big price tag, and that we would like to apply to this problem. So I think it's definitely going to be important.
Yeah, state-of-the-art electron microscope is about $6 million. Detector, a couple of $1 million. Screening microscope, about $3 million. So that's just looking at that one aspect of structural biology, and then things like creating core facilities for all these various approaches. For example, the structural core, the CRISPR technology core, cell biology core, et cetera. I mean, it's a big, because we're not just taking one approach to studying RNA, we're taking literally every approach, and the underpinnings for this are correspondingly large scale.
Well, this question follows on these comments. So if, particularly on RNA, which is so hard to work on because it's so fragile, if you accept that, if that new technology often ushers in new discovery, what type of new technology, either coming online now or foreseeable in the future, would make you most excited?
I can kind of try to. So, am I on? Okay. Okay. Can you guys hear me? Okay, so that's a great question. Thank you. We actually have one of the pioneers of this amazing new technology called nanopore technology, Mark Akeson, who asked a question earlier and who was also a Kraw lecture speaker last month. And through the efforts of his lab, we've really developed this amazing sequencer. It's called like a MinION. It's about the size of a thumb drive, and you can just directly sequence strands of DNA or RNA to discover, what are the properties of these RNAs or DNAs. And you can even look at modifications. We didn't talk about modifications at all, but these RNAs are not just naked RNA, they're chemically modified. And so through these types of technologies, we can actually look at these modifications directly. And so there's a lot of emerging technology, some of which originated at UC Santa Cruz through the efforts of Professor Akeson's lab. And also my colleague Chris Folmers, who's in the audience, he's one of the pioneers as well, working on this technology to look at single cells, look at RNA molecules in individual cells and sequencing them on this little thumb drive that you can just connect to your laptop and sequence DNA and RNA directly. And so there's a lot of these amazing technologies that are coming online through the efforts of researchers at Santa Cruz that we're really hoping to leverage to understand more deeply these fundamental properties of RNA.
All right, so there's no doubt that that's going to be an incredibly powerful sort of lens to apply to the RNA world. It's going to be really excited to see what happens over the coming years. So I think I'm getting the signal that the bar is open, I guess. So we have to sign off. Thank you for coming, and thank you for your support and for all those great questions. And we'll see you at the next Kraw lecture series. And one last thing, I want to thank George Kraw for probably being the greatest champion for UCSC that I've ever met. George wants this campus to thrive and to be excellent or outstanding in everything that it does. And it's just, it's great to meet somebody with so much energy and just passion for the university. So thanks a lot.